Come and sit. Hello, everyone. Welcome back. I hope you enjoyed lunch. I really enjoyed the posters. It was a very difficult judging situation going on. I'm sure all the judges found the same. So I appreciate that not everyone's quite back yet, but to keep the time, we'll get started. A little bit of a soft start here. First, it's my honor to present a prize for us. Unfortunately, the prize winner can't be here. My name is Spires-Jones. I am the editor-in-chief of Brain Communications. And at Brain Communications, one of our goals, as is the charity of Brain and Brain the Journal, is to facilitate career development of neuroscientists and clinical neuroscientists. And as part of that, Brain Communications instituted an Early Career Researcher Prize, where each year we have a call for nominations for the best paper published in our journal, where the first author was either a PhD student or postdoc or other early career researcher. We ask everyone who reads the journal on Twitter and all of our editorial team to nominate papers, and then we have a vote in the editorial team. And this year's winner was Stan Hrabowski for his paper, Aging and Alzheimer's Disease Have Dissociable Effects on Local and Regional Medial Temporal Lobe Connectivity. Stan's currently a postdoc at Penn at the University of Pennsylvania, studying structures of the medial temporal lobe in aging and Alzheimer's. Most of his doctoral work is centered around the medial temporal lobe, especially in vivo functional imaging of this area. And his near-term research goals involve high-resolution functional imaging projects at 7 Tesla to investigate neurobiological differences in healthy versus pathological aging. So I'm hopeful that we might have a video here. Do you know where we get Stan's video to play? Yes, okay. So Stan, since he couldn't be here, he's pre-recorded, so I hope you'll enjoy his little summary of the paper. It's very short. Maybe. Do I need to push anything else to get to play? Oh, okay. We're not hearing him.
Thank you for the invitation to present my work at Brain 2024 conference. Today, I will talk about the differential effects of aging and Alzheimer's disease on medial temporal lobe connectivity. First, I would like to thank everyone who showed an interest in our 2023 Brain Communications paper and who nominated this work for the Early Career Research Award. Here are all the disclosures of people who were involved in this work. As some of you know, deteriorating medial temporal lobe function has been linked to memory decline in both aging and Alzheimer's disease. Critically, MTL is not a uniform area, and anterior-posterior gradients in MTL function have been found in normal cognition and in neurodegenerative diseases. In humans, the anterior MTL is a part of the so-called anterior temporal network that is highlighted in orange on the image to the right. The posterior MTL belongs to the posterior medial network, which is depicted in blue. In the study, our main objective was to determine whether and how age-associated functional changes within the medial temporal lobe and in the anterior temporal and posterior medial brain networks differ from those that are caused by Alzheimer's disease. To address this research question, we examined functional connectivity, both within the MTL and within the two MTL-affiliated brain networks. Recent animal literature indicates differential role of amyloid versus tau pathology on MTL circuitry. The flowchart diagram on the right summarizes key findings from animal and in vitro studies. There is substantial experimental evidence by now of amyloid-induced hyperexcitability within the MTL, in particular in early AD. Elevated activity levels in turn facilitated tau accumulation and spread. However, as the disease progresses, accumulating pathology results in loss of synapses and neuronal death, which has a negative impact on MTL function. A 2021 study from our lab revealed greater vulnerability of the anterior MTL to tau accumulation in Alzheimer's disease. In contrast, prior cognitive and neuroimaging studies indicated that it is the posterior MTL, not the anterior MTL, that is particularly vulnerable in healthy aging. In the context of previously published literature, we had 4 primary hypotheses in the current study. First, as suggested by animal models of AD, we expected to see amyloid-induced hyperconnectivity in the medial temporal lobe during the earliest preclinical stage of Alzheimer's disease. Second, we expected this effect to moderate or to be fully reversed by the time the disease becomes symptomatic. Third, we expected to see posterior MTL vulnerability in aging and anterior MTL vulnerability in AD. Lastly, given consistent findings of reduced intranetwork connectivity in aging, we expected to see negative relationship between age and intranetwork connectivity in both the anterior temporal and posterior medial networks. To test these hypotheses, we analyzed the data from 179 participants from the Aging Brain Cohort study of Penn Alzheimer's Disease Research Center. 36 of those were cognitively normal young or middle-aged adults. 80 were cognitively unimpaired older adults without amyloid pathology. In addition, our sample included 23 cognitively normal amyloid-positive older adults with preclinical Alzheimer's disease and 40 cognitively impaired amyloid-positive older adults with MCI or early dementia due to AD. The first two groups were used to study the effects of age on MTL function, and the data from the bottom three groups were used to study the effects of AD progression. All of the MRI scans that we used were acquired on a 3 Tesla Siemens Prisma scanner, and here we used specifically a high-resolution T1 structural scan and a 2 millimeter isotropic resting state functional MRI acquired while participants had their eyes open, and acquisition parameters for this fMRI scan resembled those that were used in the Human Connectome project. MTL subregions were segmented on T1-weighted structural MRI using the ASHES T1 pipeline that was developed in our lab, and the fMRI datasets were pre-processed using fMRI Prep 20 software package. For statistical analyses, we performed connection-level GLMs with age, sex, and movement metrics as nuisance covariates. Cluster-like NBS technique was used to control the family-wise error rate. First, I will discuss our intra-MTL findings. In our intra-MTL analyses, we focused on 5 memory-related MTL subregions: the perirhinal cortex, the entorhinal cortex, the anterior hippocampus, the posterior hippocampus, and the perihippocampal cortex. To map functional relationships within the MTL, we extracted time courses from each of those MTL regions, and we correlated them with each other. Qualitatively, we observed age-associated reduction in functional coupling within the MTL. However, notably, this effect was more prominent in the posterior MTL. This qualitative observation was confirmed statistically. In particular, our statistical analysis revealed that it was the perihippocampal and posterior hippocampal connections that were most vulnerable to the effects of age in the cognitively normal amyloid negative group. Declining functional connectivity with age is a lifelong process, and it seems to begin in early adulthood. This effect cannot be fully accounted for by age-associated structural atrophy in the MTL. When we examined the effects of AD progression on intra-MTL functional relations, qualitatively, we observed 2 key phenomena. First, we saw weaker intra-MTL functional connectivity in individuals with symptomatic AD. Furthermore, we observed strengthened functional connectivity in the anterior MTL in the preclinical group. Direct statistical test for a rise and fall in a functional connectivity over the course of AD revealed hyperconnectivity in individuals with preclinical AD, and those connections were centered around the perirhinal and entorhinal ROIs. This inverted U-shaped effect could not be explained by AD-associated changes in MTL volumes or thickness. Lastly, we looked at the effects of age and AD on broader MTL-affiliated cortical networks. As we hypothesized, we observed a negative relationship between age and intranetwork connectivity in both the anterior temporal and posterior medial networks. Consistent with our intra-MTL findings, we observed increased connectivity in preclinical AD, specifically in the anterior temporal network, and affecting specifically those connections that represent direct functional coupling between the anterior MTL and its one-hop neighbors in the anterior temporal system. Together from all these findings, we draw 4 primary conclusions. First, the preclinical stage of Alzheimer's disease is characterized by hyperconnectivity in the anterior medial temporal lobe. This hyperconnectivity effect originates from those regions in the anterior medial temporal lobe which are prone to early tau accumulation. In contrast, normal aging is associated with a gradual decline of functional connectivity in the posterior medial temporal lobe and within both of the MTL-affiliated brain networks. MTL network changes in Alzheimer's disease are non-monotonic and are distinct from those that are observed in normal aging. I would like to acknowledge my colleagues at Penn Memory Center, Pixel Lab, and Penn ADRC, without whom none of this work would have been possible. I would also like to thank my advisors, David, Sandy, and Paul, who helped to guide me on my scientific journey, without whom I would not be presenting in front of you today. I'd also like to thank the National Institute on Aging and the Alzheimer's Association who funded this work. This concludes my talk. If you have any outstanding questions, please do not hesitate to reach out and contact me via the email that you see on the screen.
So I hope you'll join me in congratulating Stan on his great win. And next, Dr. Manuela Marescotti, the scientific editor for Brain Communications, will chair the Data Blitz session. So please get ready, Data Blitzers.
Hello to everyone. So the first data blitz for the first speaker is Juliette Dufour for the talk Before It's Too Late, Dynamic Changes in Sodium Neurite Fractions Uncover the Early Phase of Potential Reversible Cellular Energy Distress Preceding Neurodegeneration in Multiple Sclerosis.
So thank you very much for giving me the opportunity to be here today. My name is Juliette Dufour, and I'm a PhD student and a neurologist at the Paris Brain Institute in Paris. So the main challenge in multiple sclerosis is to better understand the mechanisms that lead to neurodegeneration, which is the main subject of clinical disability and for which we don't have treatment today. So one of the key phases that precedes neurodegeneration is actually energetic dysregulation. How? Because after demyelination, the neuron will develop compensatory mechanisms such as the redistribution of sodium channels along the denuded exolemma, and this required extra ATP needs. And this will lead, after a certain period of time, to a state that we call virtual hypoxia. And this state is really crucial for the neuron because it is a state where it suffers from energetic distress, but it's not yet dead. And so this virtual hypoxia is composed by 3 main stages. The first one is the increase in the intracellular sodium concentration. The second one is the increase in the extracellular sodium concentration due to the reverse of the sodium-calcium exchange. And the final one is characterized by irreversible neurodegeneration. And the main point here is that the 2 first phases may be potentially reversible and so represent a time window to prevent neurodegeneration. So our goal was to the first time to explore in vivo these very 2 first initial phases, potentially reversible, of virtual hypoxia in people with MS. And to assess this question, we use a combination of sodium MRI at 7 Tesla and NODI, which is neurite orientation, dispersion and density imaging derived from diffusion MRI to answer this question. So we enrolled 38 people with MS and 21 sex and age matched controls. For each patient, we did an exam with EDSS score, which is the main clinical score in multiple sclerosis. And for each subject, we defined the brain PAT score, which is the differences between the predicted age from T1 images and the real biological age. And it's supposed to be a marker of the neurodegeneration. So the first step was to obtain these total sodium concentration maps after the calibration of sodium MRI for each subject. The second step was here to compare voxel-wise control and patients to obtain pathologically increased maps of total sodium concentration for each subject. And the final step was to combine these total sodium concentration maps with the NODI maps to obtain for the first time individual profile of intranorate sodium fraction, extranorate sodium fraction, and the ratio between these two measures. And so our first key result is that we showed an increase in total sodium concentration, as you can see here, in all the regions of the brain, including the normal appearing white matter, which is the white matter outside the lesions. And so it means that it exists energetic distress in the regions that are completely looking normal to conventional MRI. Our second key result is that we found that in the normal appearing white matter, the intra and the extranorate sodium fraction was actually evolving with time. As you can see in here, the intranorate sodium fraction decreases with time, and here the extranorate fraction rises with time. And this is actually reflecting these two initial phases of virtual hypoxia that I talked to you earlier. And our third result was that we found that a higher of this ratio, extra over intranorate sodium fraction, was predictive of neurodegeneration biomarkers such as the EDSS, so clinical disability, and the brain PAD. And with a 44 increase in the probability of having motor disability for every 1 unit increase of this ratio. And more interestingly, we saw that actually this ratio becomes to be associated with clinical and radiological neurodegeneration when it exceeds the extra fraction exceeds the intra fraction by 70%. This was important because it means that below this threshold, the ratio could reflect potentially reversible energetic dysregulation, but not yet the irreversible neurodegeneration. And so, to conclude, our results suggest that individual changes in intra and extranorate sodium fractions can capture the initial two phases of the cellular hypoxia, virtual hypoxia, in multiple sclerosis, and during which this neuron damage is still reversible. And these results provide insights into the patient-specific therapeutic time window for cell rescue. And with this, I would like to thank you for sure my team in Paris and you for your attention. Thank you very much.
Thank you very much for the great talk. Is there any question for Juliette? Time for one question. Yeah.
Beautiful work. How would you explain that there's a global rather than lesion-specific increase in the biomarker that you investigated? Is that indicating that there's much more that we cannot assess on usual MRI going on, or is that something unspecific? How would you interpret it pathophysiologically?
Actually, the increase of the first step, so the increase in total sodium concentration, was completely heterogeneous into the normal appearing white matter. And so we are, for now, I showed the ratio in all the normal appearing white matter. Now we are looking in the regions that are increasing total sodium concentration to see which regions, because it's not homogeneous for sure. So it is what we are going to do now, and maybe I will show you next Congress the results.
Thank you very much. Thank you. Okay, so time for the next talk is we have Gloria Bellante. Inoculation studies in mice indicate the emergence of tau prions in traumatic brain injury patients. Please.
Okay, thank you so much. So we have previously demonstrated that a focal single severe traumatic brain injury is able to deposit pathological tau deposition in starting from the pericontusional tissue at early time point after injury. And these pathological forms of tau then spreads all over the brain, also reaching the contralateral hemisphere. And to study these prion-like properties of TBI-induced tau, we perform a transmission study by inoculating the the brain contusion from the ipsilateral cortex of a wild-type mouse that was injured 1 year post the inoculation. And we inoculate this homogenate, in the hippocampus and cortex of naive wild-type recipient mice. And after 1 year, we observed that there was a cognitive impairment, a synaptic alteration, and widespread tau pathology in these mice, thus suggesting that TBI-induced tau in the murine model display a prion-like behavior. So the next question was, is also human tau pathology transmissible? So we took advantage of the 3 different human brain contusion and from severe TBI patients that underwent to refractory intracranial hypertension. And as human control, we used the peritumoral tissue from a glioma patient, and we characterized these tissues in terms of pathological tau deposition using immunohistochemical analysis and biochemical analysis. And then we performed the same primary transmission study as before by inoculating the human brain contusion in wild-type naive recipient mice. And what we observed was that the inoculation was inducing an early and persistent cognitive impairment as assessed by the novel object recognition test up to 1 year post-inoculation. And this was associated with pre- and post-synaptic dysfunction and widespread tau pathology. But since this effect could be just a proxy effect of the toxicity induced by the inoculation of the human brain contusion, we decided to perform a serial transmission study, this time inoculating the brain homogenates from the inoculated wild-type mice. And again, we found a cognitive impairment up to 1 year post-inoculation associated with widespread tau pathology. And to assess the key role of endogenous tau in the spreading of this tau pathology, we decided to perform the same experiment, this time inoculating also the tau knockout mice. Here, yet, the idea is that in tau knockout mice, the pathological tau from the human brain contusion can't propagate and can't mediate the neuropathological function and the cognitive effect. And this is what we observed in our experiment, because 16 months post-inoculation, while wild-type mice inoculated with the human brain contusion display a clear memory impairment, tau knockout, the cognitive function of tau knockout mice was preserved, as assessed by the Y-maze test. And then we focused on the wild-type group, and we performed a gene expression analysis to assess and to investigate the neurodegenerative pathways using the neuropathology panel by nanostring. And we found some differential expressed genes in our inoculated mice that are related to lower myelination and impaired vesicle trafficking and neuroconnectivity. And importantly, we also observed that there were genes associated with activated microglia. And activated microglia was also the most enriched pathway in our inoculated mice. And so we decided to perform some morphological analysis in the microglia of our inoculated mice, focusing on the rod-like population. The rod-like microglia is a mysterious population, because its role is not well defined, but has been associated with pathological conditions such as Alzheimer's disease and also preclinical model of traumatic brain injury. And as you can see from the graph, we observed that the rod-like microglia was the number of cells, rod-like microglia cells was higher in our inoculated mice. So to conclude, we demonstrated that also in humans, self-propagating tau is induced by traumatic brain injury and shows the prion-like behavior and could explain how the initial biomechanical impact may predispose to neurodegeneration later in life in these patients. And I would like to thank my laboratories in Milan and Dublin. Thanks.
Thank you very much. Work. So, yeah, time for one question.
Really great work, and it's great to see that evolving over time. The microglial result is very striking and very kind of in keeping with a lot of the AD kind of focus on microglia. I wonder whether you could say something about the mechanism by which the microglia are influencing the propagation of tau.
Yeah, so this is exactly the work that is ongoing now, because we are performing so it's very interesting and has been demonstrated that microglia it's actively involved in the spreading and seeding of tau pathology. So now we are studying this process in TREM2 mice that are holding the R47H mutation. that is a mutation that has been associated and increases the risk of the development of Alzheimer's disease. So the study is at the very beginning, but the idea is to understand if this mutation can promote and aggravate the spreading and the pathological tau after traumatic brain injury. It would be super nice to see if soluble TREM2 could be used as a biomarker that then can be translated into clinic to see if it reflects, let's say, the pathology that we can find in our model.
Thank you. Thanks. So the next speaker is Abidemi Otaiku. And the talk is about EEG slowing increases the risk of future dementia, a prospective study of 2 population-based cohorts.
I've got a sore throat, so I'll put that up front. My name is Abidemi Otaiku, and I'm a clinical research fellow at Imperial and the UK Dementia Research Institute. I'm going to be presenting work which I've carried out over the past few months, aiming to identify EEG biomarkers that could predict future dementia in the general population. And this work will suggest that EEG slowing may be one such marker. It's been known for several decades that Alzheimer's disease and other forms of dementia are linked to a slowing of the EEG spectrum. And that includes a shift from fast frequencies, such as gamma, alpha, and beta, towards lower frequencies, like theta and delta. Recently, the slowing in Alzheimer's disease has been quantified by various metrics, one of which is the theta-alpha ratio, the proportion of theta power in the EEG spectrum to alpha power. And this is a figure from my colleague at Imperial, which has shown that the theta-alpha ratio in Alzheimer's is approximately double that of healthy controls. More recently, it's been shown that slowing of the EEG also occurs in mild cognitive impairment, or the prodromal stage of dementia, and these effects tend to be greater during sleep compared to wakefulness. But what remains to be seen is whether these changes may even precede cognitive impairment and may be a preclinical sign of dementia that occurs several years or decades before symptoms emerge. This is what we aim to find out. I used the data from 2 population-based cohorts from the USA, the study of osteoporotic fractures in men and the study of osteoporotic fractures in women. And at baseline, all participants had in-home polysomnography, which is the gold standard measure to detect sleep. The majority of participants came back over the following 12 years, and we could therefore relate sleep measures, or EEG measures, to cognitive outcomes. My colleagues and I developed a pipeline that could automatically detect EEG rhythms across 3 vigilance states: wakefulness, rapid eye movement sleep, and non-rapid eye movement sleep. So for each participant, we had 3 separate metrics: slowing, or theta-alpha ratio, which we could use in our analyses. I hypothesized that individuals who had a high theta-alpha ratio, more slowing, would be more likely to develop dementia over the subsequent 12 years. And I hypothesized that those who had a higher ratio, more slowing, would experience faster decline over the period. So what did we find? So we categorized theta-alpha ratio based on the distribution of baseline. So those who were in the lowest quartile were considered to have a low theta-alpha ratio. Those in the highest quartile had a high theta-alpha ratio, and those in the middle 2 quartiles had a moderate ratio. So compared to those who had a low theta-alpha ratio, men with a high ratio had a more than 2-fold risk of dementia over the subsequent 12 years. And in women, the effect was even stronger. Compared to those who had a low theta-alpha ratio, those with a high theta-alpha ratio had a 10-fold risk of dementia over the follow-up period. And these effects were consistent across wakefulness and REM sleep, but they were strongest in non-REM. EEG slowing predicted cognitive decline. So this figure on the y-axis is cognitive scores, x-axis is time since baseline. And as you can see, compared to those who had a low theta-alpha ratio, the blue line, those who had a high theta-alpha ratio, the green line, exhibited a faster decline over 12 years. And once more, we saw a similar pattern in the females. Blue line, low ratio, green line, high ratio, and as you can see, they had a faster decline. And once more, we found a similar pattern in wakefulness and in REM sleep. In 1,000 men from Mr. Ross who had a second polysomnography up to 8 years after the first one, we wanted to see whether EEG slowing increases with aging and whether this age effect may be accelerated by preclinical dementia. And as you can see, slowing increased over time in those who remained dementia-free, the blue line, but in those who developed dementia, the increase was more rapid, which suggests that slowing may be a preclinical sign that may accelerate nearer to diagnosis. And finally, we wanted to see whether there may be genetic basis to this association. So compared to those who had, so the higher genetic risk people had for dementia, based on ApoE status, the faster the theta-alpha ratio increased over time, which suggests that this slowing effect has some genetic basis. Conclusions. Our study has shown for the first time that EEG slowing precedes clinical dementia by several years or decades in cognitively healthy people. We've shown that a single night of in-home sleep EEG may be used to detect preclinical dementia in the population. These findings advance our knowledge of how dementia affects the brain in the early stages. And pragmatically, this study shows that EEG slowing and other markers may be used as functional measures to screen for dementia alongside genetics and blood-based biomarkers. I'd like to thank my supervisors, Professor David Sharp and Dr. Gregory Scott. I'm poster number 61, and I'd highly recommend you send me an email. Thank you.
Thank you. One question.
Hello. Thank you. Really, really lovely, crisp talk, especially within the time. I hope you don't mind me saying this, but you and I are black. We've not really had a lot of conversation about diversity here. And you've mentioned ApoE, and I wondered if you had any idea of how it might be configured differently in people from different backgrounds and how that might relate to your research. But I really enjoyed the talk. Thank you very much indeed.
So I haven't really given much thought towards those differences, but my understanding is in this cohort, the ApoE status was determined in Caucasian people. So it's possible that there may be differences amongst different racial groups. So I probably couldn't answer that. I would suspect that the differences would be similar across racial groups. That's just my suspicion.
Thank you. Thank you very much again. Okay, so the next speaker is Helena Dolphin. Please. Okay. With a talk about Alzheimer's disease, blood-based biomarkers, plasma p-tau217, electrochemiluminescence assay accurately detects CSF A-beta pathology and outperforms plasma p-tau181 in a memory clinic cohort.
Thank you. I won't win any prizes for brevity, I know. Thank you very much for having me. I am a geriatric medicine SPR from Dublin, and I'm going to present a little bit of work that we've done in our memory clinic cohort. So the background to this being that it has been recently recommended by a group based in the States and in Gothenburg that we should be starting to cautiously introduce blood-based biomarkers in specialised memory clinic services as part of the diagnostic workup of patients with cognitive symptoms only. And amyloid beta, be it on its own as A-beta 42, A-beta 40, or as its ratio, has been very limited as a blood-based biomarker because of its peripheral production. So a lot of the processes here have been focused on plasma p-tau species. So p-tau 181, 217 and 231 correspond to different areas along the tau microtubule that it is phosphorylated, and they have been, as different isotopes have been analysed in the different areas of the AD continuum. But p-tau 217 is currently the most promising blood-based biomarker for the detection of amyloid beta, and it may differentiate AD from other dementias and may have prognostic utility as higher levels are associated with future AD progression, both clinical and hippocampal atrophy. But what's quite interesting is how do we measure blood-based biomarkers? So, I'm not sure it does. This is an interesting paper that came out last year, headed up by Professor Teunissen in the Dutch group looking at this. So the gold standard for analysing blood-based biomarkers is either mass spectrometry or Simoa. But both of those measures require some capital investment because they're quite expensive, both the labour costs to run the analysers but also the machines themselves. So we were interested in looking here at a thing called electrochemiluminescence. So what is that? It's an immunoassay It's cheap, it's reliable, it does have low multiplex capabilities, but it does have high sensitivity and specificity in the right cohort for detecting the analyte that you're looking at. So our research questions. We wanted to look at what is the diagnostic accuracy of plasma p-tau217 in detecting AD pathology in a real-world memory clinic setting, and what is the diagnostic accuracy of a commercially available high-sensitivity plasma p-tau217 ECL immunoassay in detecting A-beta pathology in patients presenting with early cognitive decline. So an ECL assay involves coating your biotin antibody with the analyte that you're interested in. You boost it with a thing called turbo boost antibody. You're going to have to believe me on that one. But the most promising part of this is that it is relatively cheap and reliable to use. And the current CSF analysis uses the exact same technology as this. It doesn't require a huge amount of capital investment. It's relatively, as they say, it's relatively plug and play. And we are trying to analyse now in a real-world scenario if it is reliable. So our methods, we have set up a biobank of all of the patients who come through to our memory clinic cohort. So all of our patients who come through to our memory service are offered CSF testing, and as part of that they are offered biobanking of their samples. So we take 2 EDTA, 2 serum lithium heparin, and then we take 8 aliquots of cell-free CSF, and this is what we used to analyse and see how this assay performs in a real-world cohort. So these are the characteristics of the sample that we have analysed so far. So we have 108 participants. I'll just zone in here for you on the A-beta positive patients. The average age is 69 years old, with an average lead-in time, a duration of symptoms of 24 months, and we'll go through now in a minute the different plasma biomarker results in these patients. So this is the result of the plasma 217 ECL biomarker. So it was nearly 4-fold higher in the A-beta positive versus A-beta negative group. And we found that it had an area in the curve of 0.91, exhibiting excellent performance for detection of A-beta pathology in CSF. So what about tau? We looked at how this plasma p-tau217 analyte performed in detecting tau. It was significantly elevated in those who were CSF 181 tau positive versus negative, but the area under the curve for that was 0.83, which was significantly poorer. We also found as well that there were significant differences. There were no significant differences seen between the A-beta or the amyloid negative tau positive groups, indicating that this is a marker of amyloid positivity. And for the amyloid positive individuals, concentrations were significantly higher with those with dementia versus MCI, and that was determined by their CDR score. Sorry, no, they didn't all come through. So what then if we did a head-to-head between p-tau181 and p-tau217? So p-tau181 was only 1.7 times higher in those that are A-beta positive versus A-beta negative, and p-tau181 exhibited inferior performance than the p-tau217 analyst in the detection of A-beta status in this population, with an AUC of 0.91 versus 0.72. And we also found an interesting correlation here in plasma GFAP and the A-beta positive and A-beta negative group, with a significant correlation between plasma p-tau217 and plasma GFAP in only the A-beta positive group, but only a trend in the A-beta negative group. So this demonstrates excellent performance of a novel, high-sensitivity, commercially available plasma p-217 ECL immunoassay for the detection of A-beta positivity in patients presenting to a specialist memory service. So why is this important? We note here that we've got greater specificity than sensitivity in this group, because we can see in the assay that those that are A-beta positive still have about a 10% false negativity rate in this assay, and the potential for false negatives may be significantly higher than in a community cohort, because this nice paper came out recently to let us know that the blood tests for Alzheimer's disease are going to be particularly reliant upon the prevalence of the disease in the community, in the cohort that you perform it in. So as you can see here, the group that we have, the memory clinic cohort, may have up to 70% of underlying disease pathology, so the likelihood of false negatives is going to be higher in that group. as opposed to in a screening test in a healthy population, the false positive rate is going to be a lot higher. So these are issues that are going to have to be teased out in multiple different cohorts and explained as these come to market. Thank you. I'll take any questions.
Any question?
Stunned into silence. That's great.
Thanks very much. So I take it the amyloid positivity is on CSF?
Correct. That is on CSF. So that was both on a Roche analyzer and Fujirebio. We had them over two systems. But
So I wondered what would happen if you took it as the AD diagnosis clinically rather than the CSF amyloid positivity.
So that is fascinating. So we don't just do clinical diagnosis, apart from patients who are over the age of 80 or who decline CSF testing at the moment. But the data would suggest that we are about a third off in the literature for those who just have a clinical diagnosis of Alzheimer's disease. They don't have the underlying pathology in about a third of cases.
Thanks.
Thank you very much.
Thank you.
Thank you. Okay, the next speaker is Kirsten Scott. Yes. With a talk about blood, CSF, and PET imaging markers of immune activation predict cognitive outcomes in a longitudinal study of Parkinson's disease. After you.
So peripheral and central nervous system inflammation is well described in Parkinson's disease, and you can see in the figure on the right that there is serum cytokines on the y-axis and whole brain PK11195 and microglial activation measured by PET with a clear association between the two. We also know that there have been shifts in circulating immune cell populations that are well described on, I think I can use... In this figure, you can see data from my PhD where I showed that circulating B lymphocytes were decreased in Parkinson's disease compared to controls in the blue, and this was particularly the case for a group that were at high risk of an early dementia. And this data shows some CD4 and CD8 work done by Karen Danzer in Germany, where again they showed that the CD4 and CD8 populations were decreased in Parkinson's patients compared to controls. There are many other examples across different cell types, including NK cells and monocytes, as well as neutrophils, and establishing that these sorts of changes, some of which can be labeled inflammation, precede disease progression is key to identifying relevant targets for disease modification by immunomodulation, but also to understanding the pathogenesis. So my work was part of a cohort study led by Caroline Williams-Gray looking at neuroinflammation and tau aggregation in Parkinson's disease dementia. We recruited 40 patients and 40 controls at baseline and followed them up prospectively. You can see that patients had PET imaging using PK11195 PET ligand, which is a marker of microglial activation, amongst other things. We also did immunophenotyping using flow cytometry of peripheral blood mononuclear cells and CSF-derived immune cells. And then at 3 years post-baseline, we stratified the patients by whether they had cognitive impairment or not. And the cognitively impaired group include patients with a mild cognitive impairment and those who developed dementia. And what we found was that there were a number of differences. And what we did was have a very liberal statistical approach to start off with, and we looked at bivariate comparisons between the patients with cognitive impairment in the red and the controls in the blue, and we just took any variable that reached a significance of 0.05. And you can see there were lots of variables in the blood, in the CSF, one in the imaging, that were significant at that level. It's a combination of T cell markers, B cell markers in the blood. In the CSF, we were very interested in the ratio between the CSF and the blood because we had the thought that some of the decreases in the population we observe in the circulating cells might be explained by a shift of those cells into the CSF. And indeed, you can see across a range of different cell populations, these are the B cells, these are some of the T cells. You can see the ratios, that there is a higher ratio of CSF to blood cells in the cognitively impaired group in the red. In the imaging parameters, we took a list of 8 different brain regions that we'd identified in baseline study that was cross-sectional, and we just looked at differences between the cognitively impaired and controls in those 8 regions. And the only one that came out of that was the substantia nigra. We then took the variable in each modality that had the highest effect size. So we used standardized effect size measures of CLIF-D, and each modality had a winning variable. In the case of the blood, we had activated CD4 T cells defined by... Oh, sorry. We had activated CD4, and that's better. Activated CD4 cells defined by CD28. In the CSF, the CSF to blood memory B cell ratio had the best effect size, and we only had the one variable from the imaging to take forward for further analysis. So we had a look at these using ROC curves to try to see whether these variables on their own predicted the cognitive impairment in Parkinson's disease. And you can see that the flow cytometry variables perform quite well in that we have an AUC of 0.8 and 0.85. So this is for the CSF to blood memory B cell ratio, and for the activated CD4 cells, an AUC of 0.85. The substantia nigra microglial activation signal was slightly low at 0.79. Technically, that's considered not great, but it's close to the barrier of OK at 0.8. We then put all of these into a regression model to try to see what best predicted the outcomes at 3 years. And we found that the substantia nigra PK11195, or the microglial activation signal, was the best predictor. The caveat with that was that the numbers of patients that actually had all 3 modalities, the lumbar puncture, the PET imaging, and the peripheral blood phenotyping was lower. So when we looked at individual regression models, the flow cytometry derived variables were good predictors of cognitive outcome at 3 years as well. So I hope that you are convinced that we've shown that cognitive decline at 3 years post-baseline was predicted by an increase in the proportion of these activated CD4 cells, an increase in the CSF to blood memory B cell ratio, possibly suggesting that there's a shift of these cells into the CNS, and increased microglial activation in the substantia nigra. And so we thought that sort of suggested that interventions targeting central or peripheral immune activation, or indeed the recruitment of lymphocytes to the CNS, warrant further exploration as a disease-modifying strategy. And I've mentioned that the study is a bit limited by the numbers of patients that ended up having all 3 modalities. There's poster 53. I think the last slide should be my thanks, but somehow my first slide also fell off the deck, which is why I was a bit confused for a moment at the beginning. But I meant to say that this work was part of a postdoctoral fellowship that was funded by Brain I completed last year as part of the end of my clinical training. My only disclosure is that I'm now currently employed by AstraZeneca, but didn't present any of that work today. Thanks.
Thank you very much. Any question? Okay.
Really nice work, and really interesting kind of looking at the systemic CNS kind of interactions. I wonder whether you looked at any other events that were happening, comorbidities, during the course of the 3 years, in particular infections, respiratory infections, UTIs, anything else that might be driving that kind of inflammatory process.
So every time we saw patients at baseline, and then 18 months and 3 years, and every time they were seen, patients who had significant comorbidities in terms of autoimmune diseases or chronic infections were part of the exclusion criteria. We haven't systematically looked at the data on infections, but we did ask patients if they had infections. We have previously looked in one of our research clinic, tried to look systematically at infections in our clinic sample. And although I think our clinical experience as neurologists is that patients with Parkinson's disease get infections, they always get worse. It's harder to tease out of the data, at least the data that we've had, that the noise and the signal is hard to kind of distinguish. But I think there is kind of published data showing that, infections over time do worsen cognitive outcomes across neurodegenerative diseases.
Okay, thank you very much for the talk. So now we have Marissa Müller. Please come in. The talk is the Kind pipeline neuroanatomically, probes chronic manipulations to cortically 6B. This is very complicated for me to read, and corresponding sleep modulation. Thank you.
Thank you very much for having me. So I'm a second-year DPhil student at the University of Oxford in the Molnar, Vaisovsky and Pearson labs. And today I'm here to talk about the Quint neuroimaging pipeline, which I hope is relevant to a range of research applications here. But I'll be focusing on how it can be used to neuroanatomically probe chronic manipulations to cortical layer 6B and the relevance to sleep modulation. So this is a coronal section of a mouse brain, and the goal of the Quint pipeline is to be used to quantify the cells, the different types of cells, and the fluorescently identified combinations of these cells across the brain. My work focuses on the cortex, and if we zoom into this region, the outermost part of the mouse brain, we can see that it's comprised of historically considered to be 6 layers, and this lowest layer 6 can be split into layer 6A and the innermost layer 6B. And I'm particularly interested in layer 6B because it contains receptors for orexin, both orexin receptor 1 and 2. And orexin is responsible for promoting wakefulness and feeding. And evidence shows that layer 6B is the only cortical layer which responds to orexin. And orexin is exclusively synthesized in the lateral hypothalamic area. And the idea is that if there are subcortical signals coming up from the cortex, if the cortex plays a role in mediating orexigenic mechanisms, then it would make sense that layer 6B is a substrate for this. So we can probe these relationships by fluorescently tagging dopamine receptor 1A cells, a subpopulation of layer 6B. And we can either functionally augment or functionally silence these cells and assess what are the neuroanatomical, what are the behavioral ramifications. And pilot work has shown that there are differences in the sleep phenotype when orexin is administered in this silenced model, but no work has yet well characterized what happens to the feeding phenotype. But for now, I'm just going to focus on the neuroanatomy and try to characterize what's happening in the brain, which can then be related to behavior. So first, in order to start this image analysis, we rely on semi-automated cell detection algorithms. So this is the detection of these DRD1A cells, preliminary attempts at neuron tracing as well, and also looking at parvalbumin interneurons. Zooming out to look at the entire section, we can consider all brain regions. We can zoom in to regions of interest subcortically, the thalamic projection regions, and of course the cortex. Different regions of the cortex, different layers, the DRD1A, lower population, and then parvalbumin interneurons and other types across the brain, reconstructing in 3D and mapping to the Allen Brain Atlas. So we can repeat this across cortical sections, anterior to posterior axis. This is just one section. We take many of these serial sections, 50 microns, in both wild type and genetically modified brains. This is a schematic representation of the pipeline, which essentially consists of the imaging and pre-processing, the mapping, segmentation, merging the two, and then performing analyses. And I've done some computational modifications and refinements to the pipeline, but again, it's the same idea. We have imaging, atlas mapping, cell segmentation or detection, combining everything, and then doing reconstruction and analysis. And so breaking things down and running one sample section through this pipeline, we'll zoom in to a piece of the cortex so we can detect these DRD1A cells and also see in multiplexed analysis which ones also co-stain for complexin 3, which is a layer 6B marker, another subset. And in reverse, we can detect complexin 3 and see which ones are also DRD1A positive. And again, these are the cells that are either silenced or augmented. We do this across the brain We map it, do nonlinear refinements, combine everything together, reconstruct across multiple sections and different cell combinations. And we get lots of data out. And for example, we see there are no gross morphological changes in the areas of the cortical regions, the ventricles, or in the parvalbumin, interneuron densities, and multiplexing with VVA perineural nets. However, some preliminary data shows that there are differences in the DRD1A neurons themselves. The densities are decreased in the augmented models, and this differs across different cortical regions and layers, and this is specific to the cells that do not multiplex with complexin 3, that very tight subplate marker. When we then do this big screen, it's useful to then zoom into those regions which are implicated as being different. And you can see qualitatively that they're on the right here, there's a dramatic increase in this augmented genotype where there are these big apical dendrite arborizations. The somas are bigger, but they are less dense, and this differs across different regions. So I hope that this has given some insight into one means by which you can use the Quint pipeline, or this neuroimaging analysis method, to look at cells across the brain in a way that's cost-effective and relatively easy compared to other methods that exist. This will provide insight and comparative anatomy to different behavioral complements. And with that, thank you for your attention. Happy to take questions and come see the poster.
Thank you very much. Amazing images. So, any questions?
Thank you very much. A very nice presentation. Does your technique allow to also map more than the soma, like the axons, dendrite? It's already very nice, but for connectivity, is it possible to map these?
It's a great question, and that is the ideal next step. So the Quint pipeline in itself, as it was written, is just for soma detections. It depends on the segmentation method that you use. If you use something like ImageJ, Elastic, QPath, you can't do neuron tracing. And the Quint pipeline was designed first with Elastic. If you use something like Arrivis or Ameris, those are licensed programs, but you can do neuron tracing. And my idea is to first do the cell detections, save in data structures. I code it in MATLAB, but use structs. And then you can apply tags to each of those cells individually, map it to the atlas, and then go backwards into the data structs to then pull out the neuron tracing information. If you can do that computationally, you can then combine neuron tracing projection regions with Quint regionalizations.
Okay, thank you very much.
Thank you.
Okay, next speaker and last of this session is Zana Zoesi. A 12-year longitudinal study of sleep abnormalities in Huntington's disease. Insomnia is associated with greater cognitive deficits and disease activity.
Hi, so last one, we're almost there. So yes, I'm Zana Zoesi. I'm a brain fellow coming towards the end of my PhD fellowship with Roger Barker up in Cambridge, where I've been studying sleep abnormalities in Huntington's disease, and thank you for the opportunity to speak today. So firstly, why are we interested in sleep abnormalities in neurodegeneration at all? Well, as we've alluded a bit to already this morning, that's because there's growing evidence of an important feed-forward damaging cycle between neurodegeneration and sleep abnormalities, wherein we know across the spectrum of these diseases, neurodegeneration leads to sleep abnormalities, and we also know that those sleep abnormalities in turn lead to a host of cognitive and affective deficits. These are the very kinds of deficits that we're seeing in these patients, so it's almost certainly the case that they're contributing, exacerbating these features to some extent. But things also extend to the biological level, because we now have increasing evidence to suggest that sleep abnormalities can drive and accelerate neurodegenerative pathobiology as well. For instance, by impairing synaptic modulation, by impairing proteostasis, propagating neuroinflammation, and also by increasing levels of A-beta and tau, both by increasing production, but also by impairing this newly discovered and newly understood mechanism of the glymphatic pathway clearing these neurotoxic species during slow-wave sleep. So, of course, all this posits the notion that if we could understand and treat these sleep abnormalities in our patients better, we may be able to slow disease progression or even delay onset, making this quite a novel treatment pathway. So then why Huntington's? Why do this in a relatively rare condition? Why not do it in something more common? Well, this is because Huntington's, by virtue of its genetic basis and other features, means that it represents a very clean model through which to interrogate this field and make generalizations more widely. So firstly, because as a monogenic, fully penetrant condition, presenting in midlife, it means that we can capture individuals in the pre-symptomatic phase reliably and track them longitudinally through into symptomatic disease to try and disentangle this chicken and egg problem. And also, because onset of HD tends to be in midlife, age 30 to 50, we can do so without confounders rendered by age and comorbidities, which obviously gives us huge advantages over things like Alzheimer's and Parkinson's. Secondly, we already know from cross-sectional studies that there's a very prominent sleep phenotype in HD that begins from the pre-manifest period and shows a lot of parallels with AD and PD and other neurodegenerative conditions. And then thirdly, excitingly, we have increasing studies from the preclinical side of things showing us that if we intervene and treat sleep in HD, we can improve cognitive, motor and survival outcomes, which is, of course, highly significant given that we have nothing for this in HD that achieves this currently for our patients. And alongside that, we have the ideal translational population because, again, we can capture patients reliably at the earliest stages of disease and, again, at a relatively young age where they tend not to have other comorbidities. And yet, despite these golden opportunities, this work hasn't yet been done, so that's what I've been trying to capitalise on in my fellowship. So the bulk of my work has been around completing the first longitudinal study of sleep abnormalities in HD, using a cohort of 24 HD gene carriers, all of whom were pre-manifest or asymptomatic at baseline, and 22 controls, tracked against across a 12-year period, and at follow-up they've been stratified into those who have sadly converted to symptomatic HD versus those who haven't. And at both baseline and follow-up, sleep has been looked at through 3 objective metrics. Firstly, 2-night gold standard inpatient polysomnography, and also while they come in for polysomnography, we do serial blood sampling to look at melatonin secretion curves. But also independently, we ran a 14-night actigraphy, and this gives us a trade-off between more crude, long-term, ecologically valid measures in actigraphy versus the more accurate, high-resolution data that we get from polysomnography. And that's been looked at both in terms of validated clinical outcomes of HD, cognitive, psychiatric and motor, but also serum for neurofilament light chain as a putative biomarker of HD disease activity. So what did we find? The top line results are that the key thing that we found is that it's as HD gene carriers are transitioning into their symptomatic phase that they develop very high levels of sleep maintenance insomnia, so difficulty staying asleep during the night. And importantly, we found that that was highly correlated with psychomotor deficits and executive function deficits. Moreover, it was also correlated with neurofilament light as a marker of disease activity. And you might say, well, hold on, maybe that's just a proxy of where they are in the disease course, but importantly, there's survived adjustment for multiple confounders, including disease stage, suggesting that this may be an association above and beyond purely where they are in disease course, but a contributive factor. What's more, compellingly, the more people gained in the sleep maintenance insomnia, the worse they did at follow-up in the same domains. Also compellingly, we had corroboratory evidence from the independent actigraphy. So again, we saw evidence to tell us that there was high levels of sleep maintenance insomnia, which was again correlated with the same cognitive deficits. We also had a really nice, interesting result in terms of the melatonin profiling, namely that melatonin profiles, which should peak at 3:30 in the morning, are markedly dampened in HD gene carriers, and this is the case even more than 10 years before disease onset. And what's more, we could see from the longitudinal modelling that this falls off a cliff in that late transitional phase. So as people are transitioning to manifest disease, that melatonin loss is progressive. And what's more, we could see that the flatter the curves are, the more people are experiencing sleep maintenance insomnia, suggesting that the two may be related, as is biologically plausible. So, of course, now having mapped out this landscape of this association, of course, what we want to do is to try and intervene to see if there's causation within these associations and whether there's treatability within them. Given what I've just told you, you might say the obvious choice is melatonin, but actually, to date, the meta-analyses are not suggesting that that's going to be particularly efficacious. But we're very lucky and excited in the sleep community, as we've heard about this morning, to have 2 new sleep therapies emerging. One in the form of orexin antagonists with daridorexant licensed and approved within the UK just last autumn, and also through this new concept of closed-loop acoustic stimulation, which is a technique that's been shown robustly to be able to increase power, amplitude, and continuity of slow-wave sleep. So we are now trying to take this work through into early intervention trials in HT patients using these interventions. And if you'd like to hear more, I'm at poster 005. Thanks very much to everyone up in the Cambridge team, particularly Roger, who was ahead of his time in seeing this longitudinal study way back in 2009. And thank you to the funders, including Brain, and happy World Sleep Day.
Any questions? Yeah.
Just thinking back to the previous talk, has anyone looked in Huntington's brains at these layer 6B cells, or like what?
Not that I'm aware of. No, there's very little literature altogether. There's some really interesting data around clock gene expression in vivo, ex vivo models, but other than that, I'm afraid there's very little.
Thank you. Well, thank you, and thank you to everyone in the session. I believe we're due a coffee break, so please give a round of applause to all of our Data Blitz presenters. All right, back for 2? No, back for what time? Back for 4 o'clock, please.
Yeah.
Okay.